Okay, hi everyone. So in the following consecutive 3 lectures, we're going to learn about techniques in microbiology. At the end of the topic, the student should have a clear understanding on the various techniques involved in studying microorganisms. So in particularly appreciate the importance of accepting techniques as a basis of microbiology. And some of these parts you'll be also learning in the practicals, especially practical one. So the most important techniques in microbiology is microscopy. So the apparatus that used for microscopy techniques we call as a microscope, and it is the most basic tool in microbiology lab. So which means a microbiology lab should accommodate with at least a microscope. And then there's various types of microscope, and then the choice of particular microscopes, it depends on a few factors. So the first factor is the size of the object. So whether you want to see a microbe-sized bacteria or a nano-sized virus. So microbe size, we use a different use a different microscope. For the nano size, we have a different range of microscope. And then the degree of details you want to obtain. So whether you want to see just on the surface, you have a different microscope. Whether you want to observe the internal structure, we can use another microscope. And also the nature of the specimen, whether your specimen is in a dry form or whether your specimen in a liquid form. And also it depends on the overall purpose of the microscope objective. Again, it's go back to the main objective of your experiment. So what kind of details you want to obtain from the microscope. So types of microscope can be divided into two. So first is a light microscope and the second is the electron microscope. So the light microscope including a Brightfield microscope, fluorescent microscope, dark field microscope, phase contrast microscope, whereas electron microscopes including scanning electron microscope and transmission electron microscope. Okay, usually different lenses have a different resolving power. So resolving power of an objective lens is the ability of a lens to distinguish two adjacent objects at a particular distance apart. And usually is determined by the wavelength of the light and the numerical aperture. We call as a NA. So numerical aperture means a measure of light gathering ability of a lens. So usually higher the numerical aperture of an objective lens, so the greater is the resolution. So numerical aperture values for objective lens can be low, like up to 0.25. If it's a high power, usually 0.65. Oil immersion is usually 1.25. So usually I won't recommend you to remember this value. As long as you remember these details is sufficient for now. Okay, this is an example of objective lens. So the numerical aperture you can find on here usually. And then apart from that, another important things that you should know when it comes to the objective lens is the magnification and immersion medium. So for your practical one, you have to remember that 100 times is for oil immersion, and then you will see oil here. So which means for the 100 times you must use oil as a medium. So one might be asking why I need to use a oil immersion. So oil immersion increases the light gathering ability of a lens by allowing rays emerging from the specimen at the angles that would otherwise be lost to the objective lens to be collected and viewed. So let's look at this image. So this side is the one with oil and this one without oil. So by placing a substance such as an immersion oil with a refractive index which is equal to that of the glass slide in the space filled with the air actually, so more light is directed through the objective and a clear image will be observed. And similar thing you can observe here. So when you have an oil, so more lights are directed into the objective lens compared to the one without oil. So when more lights going into the objective lens, then you can get a more clearer image, or clearer image, I'm sorry. So this is how a bright field microscope looks like. I think for your practical one, remember for now, this is your objective lens, condenser lens, diaphragm, fine focus, and coarse focus. So when you're looking under the microscope, usually the light source from the bulb is passed through the condenser lens, will go into the specimen, and then go into the objective lens, and then it comes to your eyes. So other important components of microscope, bright field microscope is condenser, iris diaphragm, and focusing. So condenser function is to light gathering lens system, so which concentrate the light onto the specimen. So it is essential that the condenser be kept at its highest position. And the following one is the iris diaphragm. So it is located between the condenser and the light source, so which controls the amount of light that entering the condenser. So for your practical one, so please do not disturb condenser and the iris diaphragm. It's already been adjusted for you, so don't have to worry on that. On the other hand, you have to worry about on the focusing. So the focusing is divided into coarse adjustment knob and then a fine adjustment knob. So coarse adjustment is usually used to change the distance between the two objective lens and specimen very rapidly, and usually we use it to locate the specimen. So which means you have your slide, right? So your samples can be anywhere in the slide. So in order to find your sample or particular area, so we use a coarse adjustment knob. On the other hand, fine adjustment changes the distance very slowly. So usually we use to get a final tuning, so to get a sharp focus of your image. So another important thing that you should know when it comes to the microscope is the working distance of the lens. So working distance of a lens is a distance between the slide and the objective lens when a specimen in a sharp focus. So the greater the power of an objective lens, so the less it's the working distance. So for example, the low power magnification lens, so usually the distance is 8.3 mm, while the oil immersion, which is a more intense magnification lens, so the distance become smaller. So one of the most important safety precaution of using a bright field microscope is never try to decrease the distance between the objective lens and the specimen while looking down through the microscope. I think the reason we are saying that is like when you're looking at the through the ocular, there's a chances that you may hit the objective lens with your sample. So the objective lens is very fragile, so you may break the objective lens. The following microscope is the fluorescence microscope. So the fluorescence microscope is designed so that a specimen is illuminated, or known as emit light, as one wavelength, which we call as an excitation wavelength, and observed by the light emitted, which we call as a emission wavelength, at a different wavelength. So which means, so usually the sample will be excited at one wavelength, and then the reading will be done on the different wavelength, and usually widely been used in the clinical diagnostic microbiology lab. Okay, this is how a fluorescent microscope looks like. It looks quite similar to the Brightfield microscope, but then you have additionally a first barrier filter, beam splitting mirror, and then a second barrier filter. So when you turn on the microscope, the light source comes out, and then we're going through the first barrier filter. And then this first barrier filter is only will allow the blue light to pass through, which means it's only allow the blue light within the wavelength of 450 and 490 nanometer, and the blue light after that will get reflected by the beam splitting mirror, and then blue light will go into the sample, and then the sample will reflect the green light, and then that green light will go into the beam splitting mirror, and again the beam splitting mirror here will transmit the light above 510 nanometer. And then that green light will go into the second barrier filter, where the function is to filter out the unnecessary noises which comes from the fluorescence, and allow only 520 and 560 nanometer green light is passed through. And then it will go into the eyepiece, and then from the eyepiece then you can see the green lights. So at this point you might be asking like where the fluorescence comes from. So there's two ways a fluorescence can happen. Either can happen naturally, or which call as autofluorescence, or also can the cells be treated with a fluorescent dye. For example, DAPI dye. The DAPI will give a blue stain to the cells. So this is an example of a fluorescent images from a fluorescent microscopy. So usually they're very shiny, and you can see like different colors in there. Okay, the following microscope is a dark field microscope. So dark field microscope usually used to observe live organisms that are either cannot be resolved with a Brightfield microscope or cannot be stained with the standard methods. So usually the dark field microscopes have like a special condenser that focuses lights on the specimen at a oblique angle, and only light that reflects off the specimen will be seen. Therefore the organism will appear very bright on a dark background. So background usually black, and is very often used in the clinical setting for the detection of Treponema pallidum, which is a causative agent for the syphilis. Okay, this is how a dark field microscope looks like. Actually, it looks quite similar to the bright field microscope, but then the only difference is that it has like a opaque light stop, so which help the condenser to focus the light on a oblique angle. And this is the images will looks like. So if like a dark background, and then the bacteria will looks shiny or white. So this is actually a Treponema pallidum, which causes the syphilis. So the last light microscope is the phase contrast microscope. So phase contrast microscope does not require staining of microorganisms, and then also can be used to view living microorganisms. Phase contrast microscope usually consists of a special condenser and objective lens, which can help to increase the contrast of the different components in the cell. So what happens is that when the light passes through the cells, the light will be slowed down by the different refractive index of the cells, and this difference in the refractive index will be amplified by the objective lens, and this leads to the formation of a dark image on a light background. So this is an example of an image from a phase contrast microscopy. So the cells will look darker at the lighter background. Let's move on to the electron microscope. So electron microscope uses beam of electron instead of light, and then it uses electromagnetic rather than the glass lenses as in the light microscope. So usually electron microscope used to view viruses and internal structures or the details of a cells. So electron microscope can be divided into scanning electron microscope and then a transmission electron microscope. So scanning electron microscope, in the scanning electron microscope, the electron beam scan across the surface of a specimen. So the images you are getting in the three-dimensional appearance of the surface of an organism. On the other hand, transmission electron microscope, so the electron beam passes through the specimen, so which gives you the internal structure of organisms. So let's look at more details on the scanning electron microscope. So SEM, or scanning electron microscope, is very useful in studying the surface structure of intact cells and viruses. So you can resolve objects as close as 20 nanometer, so which means you can see as smaller as 20 nanometer, and the objects usually is magnified up to 500,000 times. And the scanning electron microscope also operated under vacuum condition. So in order to view the specimen under a scanning electron microscope, the specimen usually coated with a thin layer of a heavy metal, usually it's gold. So usually it's butter gold coated. So usually when the scanning electron microscope, it releases the electrons, and then the electron beam will be directed onto the surface of the specimen. Since the surface is already been coated with gold, and then the electron will be scattered from the metal coated bacteria or viruses or any sample, doesn't matter, and then the scattered electron will be collected, and then that will be converted into a three-dimensional appearance. So this is how a scanning electron microscope looks like. Usually it's large and it's taking a lot of space, and usually the room is fully equipped with air condition, because this is working with electron, is produce a lot of heat. And then this is the image will looks like. Usually it comes in a black and white because it's working with electrons. Electron doesn't produce any color. So let's move on to the last microscopy for the day, transmission electron microscope, so which is also known as a TEM. So TEM is very useful for examining different layers of a specimen. So usually used to examine the internal layer of cells and also to visualize protein and nucleic acid. And then it can resolve object as close as 1 nanometer, and then it can magnify up to 500,000 times. So as we know, electron cannot penetrate very well, therefore a thin sectioning is needed. So usually what we do, so we take the bacteria, we dry them, and then we embed in a resin, and then the resin is then sliced into a thin layers, which is like 20 to 60 nanometer. Now after that, the sliced layer will be stained to get a better contrast using osmic acid or heavy metals such as lead, tungsten and uranium. Okay, this is how a transmission electron microscope looks like. So usually it's as big as a scanning electron microscope, and also like accommodated with a fully air-conditioned room. And this is the image you get. So this is like one bacteria, and then you can see what is inside the bacteria using a transmission electron microscope. And then again, the images you are getting is 2D. Okay, the following lectures will be on the preparation of specimens for light microscopy. So there's a three-way you can prepare specimen for light microscopy, such as a wet mount method, hanging drop method, and smears. So smears is quite relevant for your practical one. So let's move on to the wet mount. Okay, so in order to do wet mount, a drop of medium containing the organism is placed onto the microscope slide. And then the slide then view under the microscope, usually used to view the live organisms. So in particular to view the microbial motility, how fast they can move, and then in which movement, which direction they move. And then this method usually is quick and simple. Okay, this video will show you like how to make a wet mount.
This demonstration is how to prepare a wet mount. The purpose of doing a wet mount is to check for the motility of a cell and also the size of a cell. Make sure your gloves and your goggles are on. I'll be demonstrating how to do a wet mount when the starting material is a liquid, and then when the starting material is coming from a colony on a Petri plate. Start by taking a slide from the box. You will need a cover slip. Cover slips are squares of either glass or plastic, and they are separated by pieces of tissue paper. When the starting material is a liquid, take a drop of the material, put it in the center of the slide. Take your cover slip, hold it at a 45-degree angle touching the slide, and gently drop it. When the starting material is a colony on a Petri plate, you will take a slide, and you're going to put a drop of deionized water in the center of the slide. Then, using aseptic technique, you'll be using your inoculating loop. You'll need to sterilize your inoculating loop by putting the loop into the flame of the Bunsen burner. Remove the lid from the plate. If there's an area on the plate where you can touch the loop to cool it beforehand, that would be a good idea. Touch the surface of a colony. Immediately put the lid back on the plate. Put the loop in the drop of water. Spread it around about the size of a dime. Sterilize the loop before you put it on the bench and turn off the Bunsen burner. Take a cover slip, hold it at a 45-degree angle, and gently drop it on the sample.
Okay, the following one is the hanging drop. So first you take a cover slip, and then you put like a tiny balls of Vaseline, or you can also put plasticine. And then after you put the plasticine, you put a tiny drop of your bacteria or the organism or specimen. And then after that, you take a glass slide, and then you gently press on top of the cover slip, which is already contain your specimen. And after that, you invert the glass slide, and then you look under the microscope. So this is how we do the hanging drop. Okay, this is another video on the hanging drop, and I'm pretty sure like the video explains much better than my explanation. So the last one is smears. So usually the microorganisms are spread onto the surface of a glass slide, and then it's go through the heat, so in order to dry them as well as to kill them.
How to make a smear and carry out a simple stain. Before staining, it is necessary to prepare a bacterial smear. Label a glass slide at one end with the culture name. Transfer a small drop of water aseptically onto the center of the slide using a wire loop. Aseptically take a small quantity of a colony from an agar plate on which the culture of interest is growing. Transfer this culture to the water drop on the slide and spread as shown here until the colony and water form an emulsion. Set down the slide and allow the smear to air dry. Once completely dry, heat fix the smear by carefully passing the slide, smear facing upwards through the Bunsen flame 3 times. Do not overheat the slide or hold the slide in the flame. The heat-fixed smear is now ready for staining. It is possible to prepare multiple slides simultaneously, providing you label all slides well.
So once you have the smear, the following procedure is a staining procedure. So the main objective of a staining procedure is to give colors to your samples to get a better contrast. So and then the staining can be divided into a simple staining and differential staining. So in this lecture we will learn about like a simple staining including a positive staining and negative staining, differential staining which including a gram staining, acid fast staining and endospore staining. So all this staining you'll be doing in your practical 1. Okay, the first is the simple staining. So simple staining, usually we use a single stain or single dye and all the cell structures stain in a same color. So it's regardless of what type. Let's say you have a blue dye and all the cells will be stained blue. And then the main objective is to reveal the basic cell shapes and cell arrangement. And then these can be divided into a positive or negative staining. So let's look at the positive staining procedure. So in the positive staining procedure, the stain is attracted to the cells and takes the color. So usually the stain or the dye is basic, which has a positive charge chromophore. So chromophore is the component in the dye that gives color. And then usually, as we know, the cell, the microbial cell, is negatively charged, and then that will attract the positively charged dye. And then the dye will stick on the microbial cells. An example of positive stains including methylene blue, crystal violet, safranin and malachite green. So which all the stains you'll be seeing in your practical 1. Okay, this diagram is to show you the mechanism of a positive staining. So usually the cell is negatively charged and then the dye is positively charged due to the chromophore. So since the cell is negative and this is positive, and then the dye will just go and attach to the cells, which makes the cells stain. Okay, this is the image we obtained from a simple staining using a methylene blue on the Staphylococcus aureus. So it's pretty obvious that Staphylococcus aureus are spherical. Okay, next is the negative staining procedure. So usually the stain is repelled by the cells and the background takes the color. And the stain is usually acidic and as a negatively charged chromophore. So as I mentioned before, the bacteria is negatively charged and the cell, the stain also negatively charged, and then they tend to repel each other. And then usually it's very useful to viewing capsules and structures surrounding a certain cells. An example of acidic stains including nigrosin and India ink. Okay, the negative staining. Okay, the cells, as I mentioned before, is negatively charged, and the chromophore also negatively charged. So therefore they excel each other, and then the dye stays on the surface or the environment. So it's make the surrounding of the cells colored. So this is an example of negative staining. So can you look here? The background is colored due to the dye, and then the bacteria is inside, and then you know the white thing are the capsules. Okay, the following one is the differential staining. So in the differential staining, we always use more than 2 dyes which react differently with different kinds of or parts of the bacteria and allowing them to distinguish. An example of differential staining including a gram staining, acid-fast staining, and endospore staining. I'm not going to go through the gram staining as we already learned in the previous lecture. Let's move on to the second staining. Okay, the second one is the acid-fast staining. And then acid-fast staining, also known as a Ziehl-Neelsen staining, and usually is used to identify Mycobacterium tuberculosis, which causes a tuberculosis, and then a Mycobacterium leprae, which causes a leprosy. And then usually the style stain with carbon friction, and then we heat the smear, and then we rinse with decolorizer with 3% hydrochloric acid, and then finally we rinse with 95% ethanol. And after that we counterstain with the methylene blue, and usually the acid-fast bacteria, which is Mycobacterium tuberculosis and Mycobacterium leprae, will appear red, while then the non-acid-fast organism will appear blue. Okay, this diagram shows the acid-fast staining. So once you got the smear, and then we put the carbon friction. So carbon friction function as a primary staining, which means that all the cells will be stained in red in color. And after that, we heat the slides, and then the heat function as a mordant to fix the carbon friction into the cells. And after the heating, we apply acid alcohol, which is function as a decolorizer. So which means whichever the non-acid fast bacteria will turn into colorless. And finally, we put a methylene blue as a counter stain to stain the non-acid fast bacteria. So this is how it looks like under the microscope. Usually the acid fast bacteria will look red, and then the background or the non-acid to acid fast will look blue. Okay, the endospore staining. Okay, endospore staining used to view endospores in the genus Bacillus and Clostridium. So these two bacteria, they can form endospore. And then maybe you can refer to your practical manual, which I will give you later for the full procedure. And then end of the staining, so the endospore will stain green, which taking up the malachite green, while the rest of the cells, sometimes which is also including the background, will stain red, which is due to the safranin. So the final staining procedure is the endospore staining. So endospore staining is mainly function to differentiate between the endospore and the vegetative cells. Okay, so first we prepare the smear, and on the smear we apply a malachite green. So malachite green is function as a primary staining, which means all the vegetative cells and endospore cells will be stained green. Now after that we apply heat as a mordant, so to force the dye go into the cells. And after that we decolorize with water. So and then this means all the non-endospore cells will be decolorized, become colorless. And finally we use a safranin as a counter stain. So finally we'll get results like this. So the cells will appear red, which taking up the safranin. The endospore will look green, which taking up the malachite green. Thank you very much for your attention. Please post your questions in the e-learning.